This is our second recording, part 2 of 2, on the cell cycle and cellular reproduction. To recap, we talked about, in our last recording, the cell cycle. It's broken up into two phases. We have interphase, mitotic phase. Within mitotic phase, we have mitosis and cytokinesis. Mitosis is the replication of the nucleus. Cytokinesis is the replication or the division of the cytoplasm. And within mitosis, we talked about prophase and metaphase. During this recording, we are going to talk about anaphase, telophase, and cytokinesis. So let's get cracking. As we look at anaphase, those sister chromatids of the condensed DNA, of the chromosomes, are going to be ripped apart at the center of the chromosome, or the centromere of the chromosome. And this is where those microtubules really earn their worth. These are really tough filaments, tough fibers that pull the sister chromatids apart. And then all of a sudden now, we have single-stranded daughter chromosomes that are going to be moving to opposite ends of the cell. And as those single-stranded daughter chromosomes, or basically just separated chromosomes, are going to the opposite poles of the cells, they're going to be pulled along and eventually put into two brand new nuclei. As those chromosomes are clustered together at the poles of the cell, or the ends of the cell, they are going to form, they'll have a new nuclear envelope form around them. The rough endoplasmic reticulum, which is an organelle we haven't talked about yet in class, and we'll talk about more when we get to that discussion on organelles. So at this point, I want you to think something else inside of the cell makes a new membrane for the new nucleus that wraps around each of the new clusters at the poles of the cell. So we have two new nuclear envelopes forming around two new nuclei at the two opposite poles of the cells. The chromosomes start to relax, and that tightly wound DNA becomes loosely wound DNA. We go from chromosomes to chromatin. The mitotic spindles that formed from the centromeres begin to disintegrate, and then each brand new nucleus starts to form the nucleoli. So as we look at this, we have anaphase. Let's back up a little bit here. We start with prophase. The chromosomes condense, the nuclear envelope disintegrates, the centromeres move to opposite ends of the, centrioles move to opposite ends of the cell. The mitotic spindles form and line up the chromosomes during metaphase of mitosis. During anaphase of mitosis, those chromosomes are ripped apart and pulled to two poles, the two opposite poles of the cell. And then during telophase, two brand new nuclei form. And if we look at the centrioles, or centromeres, depending on how it was taught to you, those mitotic spindles have left. So all of those microtubules are disintegrating. And then the key characteristic I want to emphasize for you during telophase, at least to see it on the microscope, is you start to see this cleavage furrow form. Here you can see the cleavage furrow on the photomicrograph. And then immediately after this cleavage furrow forms, we have two new daughter cells. So this is kind of a sticking point for students when we think of telophase. Telophase is specifically the last stage of mitosis, forming those brand new nuclei. But telophase of mitosis overlaps with something else in terms of time. Well, if you look at the time domain, and that thing it overlaps with is cytokinesis. So while telophase is finishing up, cytokinesis is just starting to begin. The cytoplasm of the cell is starting to divide as the two daughter nuclei are being finalized. And you can see that right here in the photomicrograph. We have the chromosomes separated, daughter nuclei are forming, and we also have that cleavage furrow of cytokinesis. Let's talk about cytokinesis in more detail. Cytokinesis is division of the cytoplasm. So I'll say this again. This is probably the fourth or fifth time I said this. Mitosis is division of the nucleus. Cytokinesis is division of the cytoplasm. So when we look at cytokinesis and telophase of mitosis, they overlap just a little bit with each other. There are some motor proteins called myosin that are going to pull on some other proteins called actin within the cell itself, the webbing of filaments or cytoskeleton within the cell. And as these myosin proteins pull on the actin proteins, it causes the cleavage furrow to form, which can be seen again right here. As we can, the cell will pinch in on itself. And this is going to eventually pinch the cell into two, giving you two genetically identical daughter cells. So, at the beginning of mitosis, or excuse me, at the beginning of the mitotic phase, we started off with one cell with one nucleus. That one cell with one nucleus separated its nuclei and then formed two daughter cells with two nuclei. And let's be pragmatic here. We started off with a parent cell that had two copies of the genome. It was diploid. And we ended up with two daughter cells that each individually had two copies of the genome. They are each individually diploid. So the two daughter cells are going to be genetically identical to the one parent cell. And that is the cell cycle. Here's an awesome YouTube video. Let's make sure it works. All right. Yep, it works.
This soccer.
But I am not going to play this YouTube video for you because I don't want to run into issues with copyright. I want my video to stay posted. So I'll put the link to that video on your course website. As we're looking at the cell cycle, there are some things that help to regulate our cell cycle. Typically, cells will divide. Cells will be encouraged to divide when they have enough cytoplasm for two daughter cells. So they make it through the gap 1, gap 2. They've replicated their DNA. They've made it through S phase. They have enough nutrients. They have enough growth factors. So these are all parts of interphase. If they can make it through gap 1, S phase, and gap 2, they're going to divide. But just because they make it through those stages doesn't necessarily mean they're going to divide. We need something to inhibit our cells from growing, and that's going to be, generally speaking, crowding them out. So if there's room, if a neighboring cell has deceased or died, and there's some room for a new cell to occupy it, there's going to be some cellular division. In properly functioning cells, we don't have our body tissues replicating out of control. So if a cell is snug, it's tightly pressing against a neighboring cell, it's generally speaking not going to divide. Or if it doesn't have enough nutrients or stimulation or growth factors, it's generally speaking not going to divide. And this is a good thing. We don't want our cells to divide out of control. When cells start to divide out of control, that's called a tumor. And generally speaking, we don't want to develop tumors, as a human being. As we're regulating the cell cycle, there's something called a molecular timer. And this molecular timer is just a series of molecules inside the cell that help to regulate the cell cycle as we go from gap 1 to S phase to gap 2 to the mitotic phase. Some of the proteins involved are cyclins. Those are proteins that are present during interphase only. We also have cyclin-dependent kinases, and these are proteins that are activated by the cyclins to add phosphate groups, hence the term kinase. Kinases add phosphates. They phosphorylate other proteins. So what do these cyclins and cyclin-dependent kinases do? They help to regulate the cell cycle. So they'll allow for us to go from G1 to S phase to G2. And they also help to regulate the stages of mitosis itself. So as we go from prophase to metaphase to anaphase to telophase, and then after mitosis to the cytokinesis. So these molecular timers, these cyclins and cyclin-dependent kinases, are proteins that regulate the cell cycle. At the beginning of the G1 phase, there's a checkpoint. And again, all these checkpoints, as we go from G1 to G2 to the mitotic phase to the S phases, these checkpoints are regulated by the cyclins and the cyclin-dependent kinases, and they check to see, are there enough nutrients in the cell? Are there enough organelles? Are there enough nucleotide subunits present so that we can replicate the genome during S phase? So these checkpoints that are regulated by these proteins, the cyclins and cyclin-dependent kinases, are where we stop and double check the work of the cell cycle. During that first checkpoint, during G1, as we're transitioning from G1 to S phase, if the cell doesn't pass through that checkpoint, it gets stuck into the G0 or G0 phase, where it can just stay indefinitely or stay for years at a time. After DNA replication, after S phase, there's the G2 phase. And as we go from G2 to mitotic phase, there's that another checkpoint. And this is where we have a lot of DNA proofreading occurring during this G2M checkpoint, where the genome is analyzed to see was it duplicated correctly during S phase. And then finally, we have a third checkpoint as we go from metaphase to anaphase. And this checkpoint double checks to make sure that the chromosomes are lined up on the midline or equator of the cell before we separate those sister chromatids. So that's all we have for the cell cycle. Make sure that you download the PowerPoint and check out this super sweet A&P video that I've linked for you. If you have any questions, please swing by my office hours or shoot me an email. Happy studies.